Hello, my name is Jewel Mitchell, and this is my exam mini video number 1. So for X chromosome inactivation, this is when one of the X chromosomes is randomly inactivated in females. This process is called lionization, and it was discovered by a scientist named Mary Lyon in 1961. The inactive X chromosome forms a dense region in the nucleus called a Barr body. These can be seen clearly throughout a microscope in females. Here's an example of the Barr bodies. We have a female cell with the Barr body represented by the arrow and a male cell without a Barr body. So for the tortoiseshell example, we also discussed this in our lecture. It's an X-linked trait because they're found on the X chromosome. So in this case, we have black is dominant, so we have a capital B, and then the yellow-orange pigment is recessive, so we have a lowercase b, and these are just the phenotypes and genotypes that we're going to use. So first we're going to start off with a male and a female for the tortoiseshell cat example, and here is the genotypes for each, one black and one orange-yellow pigment color. So we're going to start off with the males, and being that the black fur was dominant, this male would be black, and this cat would be the orange-yellow pigment color. And for the females, we have both homozygous for black and the orange color, and these are representing the Barr bodies. So it doesn't matter which the paternal or the maternal is inactivated. This one would still be black, and this one would still be the orange-yellow color. So for the tortoiseshell cat, it's heterozygous, meaning two different. So the X chromosome with the black allele would choose to inactivate and only show the orange color, and the X chromosome with the orange allele would choose to inactivate one and only produce the black color. So you end up with a cat with these spots. So long hair is dominant over short hair, and we're going to cross a yellow short-haired female to a black male heterozygous for long hair. So we have our two Punnett squares. We cross them and we get the results. We'll have 1/2 X big B, X little b, 1/2 X little b, Y, 1/2 X big H, X little h, and then 1/2 X little h, Y. Long hair is dominant. So here we use what we found in the Punnett square to make a branch diagram. And so basically we just break it down and then combine all of the different genotypes together. So the tortoiseshell cat and its mother. A cloned tortoiseshell cat would not look the same as its mother. This is because the color yellow-orange is randomly activated and deactivated in these types of cats. So if it was activated, it would be yellow-orange patches, and if it was deactivated, it would be black patches.